Hello, biology students. What we're going to do today is follow along with the Meiosis PowerPoint, the Google slide that is online, and we've already went over a couple of the slides. Like slide number 2, we went over that we have bulls, lots of bulls. We call these bulls sires because sire means to pass on your traits to the female. Well, these sires have breeds and names and birth dates just like people do, and with these sires what we want are traits out of them. So like Big Sky there on the slide on slide 2 is red and long bodied, lots of muscle linked to him, and we want that trait to add length to our calf, so we will use his genetics to be passed on. This company called ABS sells bulls genetics. One of the genetics we talked specifically about was on slide 3 about Toy Story, and how Toy Story has sired 500,000 offspring. That's 500,000 different cavies have been gotten from one bull. And Mason was the one that pointed out to us that this bull has some really kick-butt milk genetics. And so people wanted Toy Story's milk genetics. That was objective 1. The whole idea of EPDs. And an EPD is just genetics of bulls or genetics of cows. The expected genetics that we can expect out of that parent. Slide number 5 talked about meiosis is the process of special cell division that creates haploids. Haploids have half the information. So we do in the cell cycle we created duplicates. In the cell meiosis, you're going to create half of the information, so that when you take an egg and a sperm and you combine them together, you don't get double, you get a full set. So meiosis is all about reducing the number of chromosomes and producing haploids, or half the information, so that when they combine they produce diploids. Make sure you pause this if you need to go over if you need to read items and check through things. Don't just zip through the PowerPoint and the video, because we're already up to objective 2 now, creating the steps of meiosis. Slide number 7, boom, G1, S, G2, same exact thing as the cell cycle. We grow, we duplicate our DNA, so we get 2 sets of it, and then we grow again. Prophase 1 is the really funky step. During prophase 1, we have what are called homologous chromosomes. So in our picture here, these guys use your other hand. These 2 right here would be what are considered homologous chromosomes. Wow, I cannot figure this out. It's like backwards. Your brain really has to think hard. This hand, quit there, got it? Boom. These little guys here are homologous chromosomes. What that means is this is an entire chromosome, this is an entire chromosome, and these 2 chromosomes will pair up, and then they will exchange their information. So you can see that some of the information from this one moved over to this one, and that's identified here and up here and down here and up. Oh, this is hard. There. This is shown on the next slide, slide 9. It's probably a little bit better, and that whole envelope thing was just a waste of your 37 seconds. But what it's showing is that some of the information from one chromatid is moved over to another chromatid. This then causes the DNA sequence, the ABCDs, to be different. Or ABCD, sorry, AGCT sequence that's in the DNA to be a little bit different. This leads then to why we have twins that can be different, brothers and sisters that can be different, because During prophase, the information is swapped over, which then changes the DNA sequences. It doesn't change a crazy amount. That's why we still look similar, and these twins on slide number 10 look similar. It's just there's a little bit of difference. And this little bit of difference causes genetic diversity. And you better be thinking this is kick butt, because this is what makes you not the same as your parents. Because if your parents made identical haploids, you would be cruising around just like your parents are, because you would be having the same genetics that they produced. Instead, though, they produce haploids that are crossed over, so they don't have the exact same sequence. If you look on the next slide, slide 11, it shows you that metaphase occurs next. So we've got these chromosomes. These chromosomes paired up, and then they switched over their information to make it so that their DNA sequences aren't the same, causing genetic diversity. They pair up, and then they line up in the middle in metaphase. Then these homologous pairs be pulled apart and get turned into singles and get stuffed into a nucleus in telophase. This should sound very similar to mitosis. However, on slide 12, the big difference happens. We just go through another splitting. We didn't go through S, but we did go through another splitting. We go through prophase, we go through metaphase, we go through anaphase, where our no longer homologous pairs, just the singles now, are going to be split apart. We were pairs, and we split the pairs apart in meiosis 1, but this time we're going to split our singles apart. This then makes it so that there's a half amount of information in each one of the cells, called daughter cells, in telophase 2 in cytokinesis. That means that all the information is split down into a half. It was duplicated back in interphase, but then it split once in meiosis 1, and then it split again in meiosis 2. This takes us to our last objective about comparing meiosis and the cell cycle. When you look at my little charty chart there, that little charty chart gives you that the meiosis produces different types of cells. Cell cycle produces identical. Crossing over occurs. You guys can read the rest. It's trying to explain to you that this is how we get genetic diversity. If we only did mitosis, we would be getting very, very, very similar to identical cells. So by doing meiosis and fertilization, we get a lot of variance in our offspring. To revisit back to Dream On, then, Dream On is $100 per straw that you put into a female cow. You are banking on the fact that not too much crossing over occurred in Dream On semen. And the majority of the time it doesn't. That's why we pay so much money for the semen, because there's a little bit of crossing over, which is good, because then we can actually get a chance of getting even better offspring than what the male can, than what the dad or the sire had. Slowing down and reviewing that. So meiosis has a special step called crossing over. Crossing over changes the DNA sequence. It can change the DNA sequence to where it's very similar but a little bit different than the parent. If it changes it to the better, that's what we created here in the class, and as I look around through this video, you guys would be prime examples of the better. Just like with our livestock, the offspring could be prime examples of the better. It's hard to do, but we do get better. So hopefully you followed through this video, followed through the Google slide, and did your Google little slide notes, and you guys have a wonderful weekend, and we'll revisit this stuff on Monday.